Hi, everyone. This is Marvi Valcourt, and I'm the marketing manager here at UCLA TDG. I want to welcome you to today's webinar on the COVID-19 Spotlight Series based on clinical research. And I want to remind all the attendees as you come in that you are welcome to use the Q&A function on your screen of your webinar. So you can use that Q&A function to go ahead and post questions, and we will do our best to present them to the panelists. And with that, I'd like to introduce Mark Wisniewski, Senior Director here at UCLA TDG. And I'd like to welcome all my panelists to please unmute yourselves now. Thank you.
Thank you, Marvi. I'm Mark Wisniewski, the Senior Director of UCLA Technology Development Group in Biopharmaceutical Licensing and Strategic Alliances. TDG serves as a gateway between our innovative academic research, the biopharma industry, and entrepreneurs. And really our goal is to facilitate collaborations to drive new patient therapies, whether it's sponsored research, licensing, enabling new startup formation and accelerator programs. Overall, our goal is to advance the LA biotech ecosystem. Today, I am glad to present our panelists for our third LABEST Presents COVID-19 webinar focused on clinical research. And in the spirit of our annual LABEST conference, the goal of this event is to promote Los Angeles as a center for bioscience innovation, showcase the leading research and clinical research through our prominent investigators who are participating today, in order to foster partnerships between academic institutions, the investment community, and biopharma industry. And on that note, I welcome our panelists, and we'll turn it over to Marvi.
Thank you. So we're going to start with introductions from all our panelists, starting with Arash.
Hi, I'm Arash Naeim. I'm the Chief Medical Officer for Clinical Research for UCLA Health and an Associate Director for the Clinical Translational Science Institute.
Hi, I'm Judith Currier. I'm a Professor of Medicine and Chief of the Division of Infectious Diseases at UCLA.
Hi, I'm Manish J. I'm the Division Chief for Pediatric Allergy, Immunology and Rheumatology and an Associate Professor here at UCLA.
And good afternoon, I'm Alyssa Ziman. I am the Laboratory Director for the laboratory at Ronald Reagan UCLA Medical Center and the Chief of the Division of Transfusion Medicine at UCLA.
Thank you, everyone. So we're going to start with overviews from each of our researchers on the work that they're currently doing.
Great. So I'm going to talk about the COVID clinical research infrastructure. And if you can advance to the next slide. So when UCLA tried to rapidly respond to the pandemic, we felt it was important to really concentrate in three big areas. The process of being able to do the research quickly, what sort of sources we needed to be able to provide materials to our researchers, and other related things in terms of innovation that might be important in doing clinical research, both for COVID patients as well as continuing high priority non-COVID research. Next slide. So one of the areas is governance. We created a COVID oversight committee, largely also as a function of managing all of the task forces that are critical for evaluating scientific prioritization and feasibility, but also as a pathway for researchers to get access to donor funding and other pools of resources that might be available. A clinical research task force that Dr. Currier and I co-chair together, a basic science task force, and a newly created health equity task force that emphasizes really ensuring that the research activities that we do are being responsive to the diversity of the greater LA County area. Next slide. So, of course, our first goal was to try to rapidly open clinical studies as fast as possible. And in an effort to do that, which for us ranged anywhere from opening up clinical trials in 3 days up to about 14 to 15 days with an average of 8 activation days, was trying to parallel process all of the key elements required for study activation for a clinical trial. Some of these are traditional issues like contracting, navigating FDA issues, budgeting, IRB or reliance mechanisms, coordinator support, but in addition to other sort of newer issues that we would need to navigate, like electronic consent, orders within our electronic health record, remote monitoring, staffing, given the fact that we're trying to reduce the number of individuals that come on site, as well as the protective equipment that would be needed for the staff that are supporting these trials. Next slide. So, once we had our strategy for the clinical trials that Dr. Currier will speak to more elaborately in the next part of this presentation, we wanted to make sure we were collecting all the data on the patients that are coming through UCLA. Our goal is to not only have that data available for our own researchers, but in collaboration across the UC system through UC Braid, but then as part of national and international collaborations. And a lot of that needed to focus on how we identify the patients through our electronic health record, which is EPIC, and then abstracting some of the key elements that are really important to get right in our data set. We kind of standardized on the WHO core ECRF, as well as some additional high priority domains that our UCLA investigators thought were important, and then working with the health system to also bring in efforts to do symptom surveys in this population through quality improvement in population health. Next slide. And in tandem with the data was really trying to make sure that we could have a biospecimen strategy. This strategy involved collecting leftover clinical specimens on the outpatient and inpatient side, the remnants, as well as research-directed inpatient specimens, specimens at the time of screening for convalescent plasma, autopsy specimens, and then specialized programs around maternal fetal transmission and programs around healthcare workers. And these specimens are used for a whole variety of studies, a small portion of which are listed in the box to the right, but it just shows you the wealth of what's trying to be achieved through the collection of biospecimens, and largely in collaboration with large academic networks, because we need to pool the patients that we have with what's available across multiple other academic institutions in order to be able to have a sizable cohort to be able to make important discoveries. Next slide. And then in terms of innovation, in terms of clinical research, clearly one of our biggest interests, as I mentioned in the health equity comment, is trying to figure out how we can empower other local and county hospitals to start doing more clinical research in COVID. This is often a challenge because they may not have the infrastructure that we have available at UCLA. Novel recruitment techniques in terms of creating registries for new patient diagnosis for covered patients and healthcare workers, the use of our patient portal system in terms of identifying newly diagnosed patients and making them aware of clinical trials that they may be candidates for, and social media, which we use quite successfully in the convalescent plasma sort of space that Dr. Ziman will speak to later on in the presentation, as well as a need to do more remote management of trials in terms of electronic informed consent, providing access to remote monitors from our partners in the pharmaceutical industry, management of oral drugs, and remote monitoring of patients, which we're investing in terms of pulse oximetry or other sorts of things that we can do while the patient's at home. So, the challenge of COVID has provided us with an opportunity to come all together and kind of fast move on some of the innovations and demonstrate what we can do when we all work together. And with that, I'm going to pass it off to Dr. Currier to talk about the clinical trials program at UCLA.
Thank you very much, Arash, and welcome everyone. Thanks for joining us today. I'm going to just provide a brief overview of the types of clinical trials we're doing and some of the details, and then the subsequent speakers will drill down into a little more detail. But I think when we think about the spectrum of clinical studies in COVID, it's important to look across the whole spectrum of disease, from observational studies to prevention, both post-exposure prophylaxis, and then early treatment. And then I think the main focus that we've had appropriately has been for the treatment of the most severely impacted patients in the inpatient setting. Next slide. So when you think about when a person gets exposed to the SARS-CoV virus, there's a period where they may be asymptomatic. Then there can be an initial mild infection, and then some go on to severe infection, which you'll hear a little bit more about, and then others resolve. And we want to intervene at different points, and I think it's really critically important to recognize that interventions may work better when they're given earlier, and we may need different or combinations of approaches in the more severely ill patients. So as we learn about the spectrum of the disease, we need to think about what kinds of studies we're going to be doing. Next slide. So I'm breaking this down. So for observational and prevention trials, one prevention trial that got started pretty quickly here was looking at post-exposure prophylaxis with hydroxychloroquine compared to a placebo. And this is a completely remote study funded by the Gates Foundation that's still ongoing. We're really excited about opportunities to participate in vaccine trials and also prevention with neutralizing antibodies, monoclonal antibodies, and those are coming soon. In terms of observational trials, while these aren't evaluating an intervention, I just want to highlight the incredible work that was done to very quickly stand up a healthcare worker surveillance project at UCLA that is prospectively monitoring healthcare workers with both testing for asymptomatic disease and for development of antibody. And then also the cohort for collection of samples on patients that are hospitalized. Next slide. In terms of early treatment, we have an NIH-funded trial looking at azithromycin and hydroxychloroquine compared to placebo in early disease. And I think that despite all of the hype in the media and all of the politics around this, this is a potential treatment for early disease that still deserves to be evaluated in a placebo-controlled, well-conducted study. We are going to be opening a study for convalescent plasma, and I'm going to let Dr. Ziman tell you about the incredible effort at UCLA to develop our convalescent plasma program. And then through a really incredible partnership at NIH called the ACTIVE Partnership, which is a public-private partnership between industry and academia and FDA, we will be participating in a platform trial to evaluate monoclonal antibodies for treatment in the outpatient setting. Next slide. Now, when you talk about outpatient treatment for people with symptomatic COVID, one of the challenges we face is where exactly do you think you're going to do that? Because of the concerns of having people who are symptomatic brought into an outpatient clinic, and how can they be safely seen and evaluated without exposing staff and others. And so we're in the process of procuring a few of these pod units to create our own outpatient clinic for the treatment of symptomatic early stage COVID disease. Next slide. In inpatient is really where our main focus has been. And this was so critical because of the severity of illness and the absence of any known treatments. And this is a list of 12 studies. There are actually, some of the studies have two different versions, a mild disease and a severe disease. And I'm not going to read through these all, but just to highlight that there are trials that are focused on antivirals, remdesivir, when we were a site for the NIAID trial led by Dr. Yang here at UCLA. Studies looking at anti-inflammatory agents, CCR5 inhibitors, and other novel agents. And as Dr. Naeim illustrated, we were able to get these studies up and running very fast, and to the point where we have, fortunately now, as our census is dropping on the inpatient side, competition between the studies for the patients that are at UCLA, or I guess the other way to look at it is a lot of different choices and options for people, which is a really nice thing to have, and hopefully we'll be learning more about the efficacy of some of these compounds. Next slide. So just to summarize really across the spectrum, in red are the studies that are currently enrolling, in black are some that we've completed our involvement in, and then yellow are some that are still coming down the line. So we hope to be involved in COVID therapeutics for as long as it takes to figure out what's going to be effective. And we're really hoping to contribute across the spectrum of disease, both for prevention as well as treatment of mild, early and moderate to severe disease. So thank you very much. Turn it over to, oh, I'm sorry, one more slide. A couple of lessons learned. I think you heard from Arash about the speed at which we were able to get going, and that really is because of the dedicated infrastructure that we had at UCLA. And also, I think because we have investigators who are able to take on running these trials from our pulmonary and infectious disease and our colleagues in the blood bank, which you'll hear made it all possible to be involved in convalescent plasma. And I think what really made the day was the collaborative spirit at UCLA with everybody working together. Thank you.
Thanks. Okay, I'm going to go next. Again, I'm Manish J, and I'm an immunologist, so I focus a lot on how the human immune system responds to infections like COVID-19. And from the slides that you just saw, many of the treatments that we're using in the inpatient and outpatient setting focus on the immune system. Many of the treatments actually directly target the virus, but the others focus on trying to suppress the immune system to try to keep it from being overreactive, which is a major cause of complications in this particular infection. There's another side to the immune system too, which is how to try to beef up the immune system to try to prevent these infections from taking hold. And that way of thinking is very common and very well known to all of us. We use vaccines for that purpose. We try to get folks vaccinated to produce memory in the form of immunity, B cells and T cells making antibodies, and trying to protect people from getting infected before the infection spreads. We try to divide that kind of immunity into sort of two broad camps. Active immunity, where the body makes its own protection after an infection or after a vaccination, or passive immunity, where some protection is given to a person in the form of a drug or a treatment, and it may not last for a long time, but it provides a surrogate, a sort of similar role to what the body could naturally make in the form of antibodies. So passive immunity is going to play a very important role in terms of therapeutics over the next few months while we're waiting for definitive treatments to come about. And I'm going to spend a few minutes talking about passive immunity and some of the strategies that are being pursued right now. So next slide. Before jumping into passive immunity, I'm just going to mention, and Judy mentioned just now, that there are vaccines being developed. There's great websites out there now tracking some of these vaccines in development right now. As of today, there are 150 vaccines being developed in various forms around the world. As far as I can tell, 9 of them are in phase 1 trials, and 1 of them is in a phase 2 trial. Most of these, as you can see from the list, and this list came from the Milken Institute, which is tracking the vaccine development. The Gates Foundation has a similar website. Most of these are being developed in China, but a few are being developed in the US, like the one at the very bottom of the list, the Moderna one, which got a lot of press in the last few days. So we have a lot to look forward to in terms of active immunity and many, many different ways of making vaccines, everything from, as you can see on this slide, viral vectors, DNA, inactivated virus, proteins from the virus, and RNA-based vaccines. Lots of different new strategies for developing active immunity. Next slide. Okay, so the main strategy in terms of passive immunity is something called hyperimmune globulins. These are antibodies that are obtained from individuals who have already gone through the infection. And you're going to hear from Alyssa in a few minutes about how that's being done at UCLA. But when those antibodies are purified from the plasma of patients who are infected, they can be turned into a product that can be then infused into patients as a treatment. And just as an analogy to the idea of the hyperimmune globulin for COVID-19, I wanted to share with you that there are many products on the market right now that are hyperimmune globulins for other infections. For bacterial infections like botulism, the first product there, Baby Big. For viral infections like hepatitis and cytomegalovirus, the next 2. Rabies is down at the bottom left, tetanus, a bacterial toxin, and chickenpox, Verazig on the bottom right. So many hyperimmune globulin products are available today commercially. And the reason that this strategy is going to work so well for COVID-19 is because the manufacturers of these products have already gone through all the work of how to purify immunoglobulin products from patients who've already been exposed to make a concentrated antibody product and be able to deliver it to patients effectively. These same companies that are commercially making these products have done similar processes for the 2009 H1N1 influenza and also for the 2014 West African Ebola outbreak. So their infrastructure is poised for this kind of product. Next slide. So who's making these hyperimmune globulin products for SARS-CoV-2? Currently no one. There are now two major teams that are companies that are working on this. One, which is a coalition of virtually every large immunoglobulin manufacturer in the world, CSL Behring, Octapharma, Takeda, some of the biggest ones, and then others listed there, have all formed something called the COVID-19 Plasma Alliance. And together they are manufacturing a single product that will be used in a variety of different trials and eventually clinically. And the second team is a standalone immunoglobulin manufacturer named Grifols. Grifols is one of the first companies in the world to actually manufacture immunoglobulin products from humans, from human plasma. They are developing their own hyperimmune globulin product, and both of these will be available in the United States in a variety of trials starting in the next few weeks to months. So hyperimmune globulin is made, as I mentioned, from patients. Their plasma is collected, and Alyssa will talk more about how we're doing that here at UCLA. It's tested to make sure that there are actually antibodies present and there's no infection present. And critically, then there's a manufacturing process where these products are the antibodies are purified out of the plasma and highly concentrated, at which point they're packaged into vials and shipped all over the world for delivery to patients. Companies like Grifols operates facilities where they can extract plasma from patients very efficiently. There's a photo at the bottom here of a facility that's located near Bellflower, so very close to where we are right now. They have dozens, if not tens of, maybe all close to 100 chairs where patients can lie down, get an IV placed, go through an apheresis process, and have the plasma extracted. These companies pay patients for their products. You can see they pay about $50 for conventional plasma donations. They're going to be paying about $100 for every COVID-19 patient to sit in the chair and donate plasma. And patients can give twice a week. So there's an opportunity here for people to really share their plasma as a treatment for others. Next slide. Okay, so who's going to benefit most from hyperimmune globulin? I think there's going to be lots of opportunities to use this product to treat patients. Right now, I see sort of two big camps. In terms of pre-exposure prophylaxis, certainly some people who would like to get protection may include healthcare workers and first responders and people who work in nursing homes and skilled nursing facilities. Many of those folks have high potential exposure to the virus, and until there's a vaccine, many of them could benefit from pre-exposure prophylaxis. There are also patients who can benefit from this product even after there's a vaccine because they may not benefit from a vaccine. So those are patients like the ones I take care of clinically, patients who have immune deficiencies, inborn errors of their immune system where they can't make antibody immunity because of a variety of genetic mutations that prevent their B cells and T cells from going through the process of making antibodies. We follow over 400 such patients at UCLA. There are also patients who have received therapies, perhaps for their cancers, like rituximab, where they won't be able to make good antibody responses. Most of the patients who are receiving CAR T-cell therapies for B-cell lymphomas also fall into this category. They can't make really good antibody responses. Patients who've undergone bone marrow transplant and solid organ transplants also, because of the medicines they're on or because their immune system is still developing after these procedures, they will not be able to make good antibody immunity. And then there's other groups too who may benefit. Newborns, especially premature infants, have shown some benefit from hyperimmune globulins in other contexts, and potentially even elderly patients who don't necessarily make good antibody responses to vaccines could benefit from this passive treatment. This is basically giving patients antibodies because they can't make their own. So next slide. So the last area of passive immunity that I wanted to talk about is something monoclonal antibodies. So these are antibodies. They're not coming from patients, though. They're coming from a manufacturing process where companies have, through a variety of mechanisms, found or developed or brewed up in their labs antibodies that are able to block the virus from actually engaging and entering into the respiratory tract. And most of the major companies that you've heard of, pharmaceutical companies, are working on these kinds of monoclonal antibodies. There are a handful of companies that have already started clinical trials. Lilly has been working with a company called Epsilera that has now made a candidate product, and they've already started infusing patients in clinical trials at Cedars and at NYU. Regeneron has made a cocktail of antibodies, and they're going to be starting their first clinical trial in the next few weeks. And then a number of other companies. There's a group in South Korea that has already developed monoclonal antibodies for MERS and influenza, and they're developing a product for this, for SARS-CoV-2. Vir and GlaxoSmithKline has 2 products that they have developed and shown that its utility in a laboratory culture, but they haven't started clinical trials yet. And onward and onward, you can see many different companies on that along the side of the slide there that show the names of these companies, and all of them have had press releases or announcements in the last few weeks about how they're successfully developed monoclonal antibodies in the lab, and they're going to be moving towards clinical trials sometime this summer or fall. The reason this is a useful strategy is because convalescent plasma is great for big medical centers like ours, where we can get these patients and get products back to the patients who really need them. And hyperimmune globulin certainly is going to have a little bit of lag time to develop. It requires patients who had the infection to have their plasma collected for a manufacturing process to occur and for the product to be shipped back. Monoclonal antibodies represent another form of passive immunity, another product that can be delivered in a vial and stored in a refrigerator and used as a treatment, and in some cases even as a prevention. So all in all, I think there's a lot of activity, as was mentioned by both Arash and Judy. There's a lot of activity in terms of developing products for this infection. I'm very optimistic that many of these products are going to be helpful for patients who don't have good antibody immunity, and for patients who can develop, who can get some protection from infection until we actually have good working vaccines.
Good afternoon. So I'm going to talk specifically about our UCLA Convalescent Plasma Initiative. My name is Alyssa Ziman, and really speak to what my colleagues have already spoken to, and the collaborative nature of UCLA and our ability to fill this gap until more definitive therapies become available for patients with COVID-19. And again, I want to emphasize just the collaborative nature of our institution between the medical centers, our Clinical Translation and Translational Science Institute, and in my case, the Division of Transfusion Medicine. We were able to rapidly and successfully launch this initiative within 4 weeks of receiving guidance from the FDA on specifics for convalescent plasma. Next slide, please. So what is transfusion medicine? Most people don't necessarily know we exist, but we are in the Department of Pathology and Laboratory Medicine and have interactions both on the clinical side as well as in the laboratory. We're made up at UCLA of 3 distinct areas. We have our donor or our blood donor center called the UCLA Blood and Platelet Center, which really is a public-facing aspect at UCLA Health. And it allows us to engage the community. We engage approximately 25 to 30,000 individuals every year to help us collect blood products to support patient needs at the medical center. And then from there, from the products that are collected, and I showed the stats here just to give you an idea of the volume of interaction, both again with the community as well as in support of our patients. Products that are collected go on to our component processing laboratory, where they're then further manufactured into products for transfusion, and then they are provided to our Chung transfusion service to support patients. And we transfuse anywhere from 55 to 60,000 individual products annually to patients at UCLA Health. Next slide, please. So what is convalescent plasma? You've heard a lot from my colleagues about some of its uses, as well as the ability to even concentrate convalescent plasma to make a further manufactured hyperimmune product. But what we start with is an individual who was ill with COVID-19, made antibodies of their own and recovered. And from that recovered individual, we're able to collect a blood product, a plasma product that is rich in these hopefully virus neutralizing antibodies. And so our initiative aimed to recruit recovered patients and then screen these individuals both for resolved infection as well as standard blood donor eligibility, and then put them through a collection process. Again, we did this within 4 weeks, or started this program within 4 weeks of receiving guidance from the FDA, and have since, in terms of recruitment, recruited almost 3,500 individuals through a public-facing portal through our donor center, and then gone on to screen them. We have collected plasma from almost 100 individuals and collected nearly 250 units that we have used for patient care. Next slide, please. The schematic here really shows the entire process. So I just spoke to collection, which is on the left side of the image, and then we'll focus a little bit on treatment, which would be 4 and 5 of the image that is provided. And a unit of convalescent plasma looks just like a regular unit of plasma, which you can see in the picture on the right-hand side. And so as Judy spoke earlier, we have been treating patients who have severe disease through the Mayo Expanded Access Program, and we've treated 39 patients to date at UCLA. We've also provided 46 units to other hospitals throughout Los Angeles, including UC Irvine, as well as UC Davis, so Los Angeles, Bakersfield, and the Orange County areas. And in the future, as we build our inventory, we will use these units for prophylaxis and mild disease, again, as Dr. Currier spoke to earlier. And our goal will be to have an inventory that both meets our patients' needs as well as patients in the community. And I think that is a great opportunity for UCLA to expand its collaboration and support other institutions and our larger community of patients. So with that, thank you very much.
Okay, just unshared the slides now. So thank you all of you for giving us summaries on what you do. And we'd like to continue the conversation. I know, Alyssa, you said there's been a lot of collaboration. You know, we're a big institution here at UCLA. We have a lot of advantages to collaborate. But how does that happen? Like you said, we were up and running in 4 weeks, which is really a short amount of time. So how does a big institution that's, you know, in their own departments, each of you are doing your own thing, how do you come together and like put something that, you know, that complicated together? You made it seem so easy, but I know there was a lot that goes into it. So I'd like the panelists to talk a little bit about that.
I can address at least part of that, which is that, you know, it's not that we just started putting the infrastructure into place right when the pandemic had started. The UCLA Health, the School of Medicine, the Clinical Translational Science Institute, the Cancer Center, and other sort of clinical research units have been working over the last many, many years, maybe half a decade, in terms of strengthening the clinical research infrastructure at UCLA. So, you know, all of those things paid off, at least for us, when the pandemic started, in that we had a lot of the infrastructure in place that we could leverage, and, you know, it allowed us to be able to respond pretty quickly.
And then I would add that, you know, this was a unique time where people really wanted to participate. They wanted to help, and the individual efforts from people across the health system, whether it was in the laboratory, in our donor center, in the research institute, to step up and help out, even if it wasn't their area of expertise. People wanted to find a treatment for patient care, and I think that's also evident in the community and the outpouring of people who want to donate convalescent plasma, who want to just do something to help support their neighbor.
Yeah, and
Oh.
Oh, no, she froze.
I think Judy's having a little bit of technical problems there. Manish, do you have anything to add?
Yeah, I would say,
Diamonds.
yeah, let Judy talk. I think she just
Oh,
came back.
Judy, you froze a little. Go ahead.
Sorry, I was just going to say that we had some previous experience with influenza and convalescent plasma, and so that helped pave the road a little bit. But it was clinicians from transfusion medicine, you know, and pediatrics and adult and surgery. I mean, really everyone working together. Some people whose regular jobs were not as active as usual were able to completely give their time to working on this. And it was a round the clock, 7 days a week to get this off the ground, but it really has been very gratifying to be able to offer this to people in the hospital.
I would just add one other thing about bringing together the community. As much as the clinical side definitely has come together, there's also a number of basic science departments that have jumped in to the fray to try to contribute to the COVID research work, including groups that I'm participating with in computational medicine and the basic science departments of immunology and microbiology. Just crazy numbers of scientists who said, I'm going to put what I'm doing aside and work on this. I think it's really inspiring.
Yeah, that's very, very inspiring and pretty amazing. We're having some questions come in from our audience, and I want to introduce Daniel Mayhew, my colleague, who's going to pose a question or two to the panelists.
So we're seeing some interest, a lot of interest in the plasma therapy. One of the questions that came up was, of the 39 transfusions to date with convalescent plasma, can you comment on their mix, severe disease, et cetera, and results?
Yeah, I can start, and then maybe Dr. Ziman can comment. So the expanded access is considered emergency use of convalescent plasma. So it's only allowed to be used for people with severe or life-threatening disease or at high risk of progression. So it's for the sickest people in the hospital that we're able to use the convalescent plasma until there's more data from controlled trials about its efficacy in other settings.
Yeah, and just to add to that, we're participating with a nationwide group. So they have to date enrolled, I think, almost 20,000 individuals across the United States to receive plasma as compassionate use. But as Dr. Currier mentioned, until we have controlled trials where we can compare plasma that has high levels of antibodies versus standard plasma, we won't really know whether or not this is a valuable therapy.
We have another question that asks, what is the clinical opinion about decreasing virus load during active infection?
Yeah, I can try to start with that one. It's a great question. First thing is really, how do we measure virus load? Those of us who work in HIV are used to having a great measurement of the virus in the plasma. But for COVID, it's different. We're measuring viral shedding from the nasopharynx. And during the course of the disease, where the virus is may be evolving. So I think that while it would make sense that having a higher amount of virus would be portend a worse course, quantitative assays for virus measurement are not yet standardized. But there are a lot of people working on that, and I think that we will be learning more about this in the future.
Any other thoughts?
Maybe you could mention that remdesivir, one of the drugs that's being studied, is a way of reducing viral load.
Yeah, I mean, it's an antiviral, but it didn't actually reduce viral load in the studies that were done to date, but they were treating people with more advanced disease. And so I think there is going to be more data coming out of the completed study. But it may be that early in the disease is when you see the highest levels, but then the damage can be done and the disease can progress despite the fact that the virus levels have gone down. Now, on the other hand, we're still seeing people with positive PCR weeks out from infection, whether this is remnants of the virus or actual infectious virus, I think remains to be seen. But so the measurements of virus and correlating it so people can be better and still have the virus from the nasal swab. So it's not a complete picture just yet.
Daniel, I think you're muted.
No, I'm muted. I'm trying to, there's a question I wanted to ask, but it has some, so this is a question that sort of relates COVID with HIV. And the question is posed to Dr. Currier. There seems to be a phenomena of sorts occurring in the HIV positive community. I'm HIV positive. Health centers around the country, specifically Zuckerberg in San Francisco, large HIV practices in New York and in fact Los Angeles are not seeing statistically significant numbers of critical ill HIV COVID patients, albeit those numbers are under the age of 70 or 80. Is there something occurring in this population that's somewhat shielding from critical illness?
Yeah, this is a really great question. Thanks for asking it. So, we all were bracing at the beginning of COVID as to whether the disease was going to occur more frequently or have a more severe course for people living with HIV. And anecdotally, we just are not seeing that. I think that we'll start to see some controlled data from really prospective studies to try to put that into better perspective. But either it's there's some protective effect of the immune system in HIV not being overly responsive to the virus, or there's some interaction with medications that are used to treat HIV. And in fact, one of the first drugs that was looked at as an antiviral for SARS-CoV-2 was lopinavir/ritonavir, the protease inhibitor. To date in studies, it's not shown a dramatic effect. But I think there's still interest in looking at whether there's any protective effect of any of the other commonly used drugs. So I would stay tuned for more on this really interesting topic. And it's really fortunate that at least in this respect, COVID has not had a disproportionate effect on people living with HIV.
So I think one of the ubiquitous questions that are just being asked, just generally speaking, is is there a presumptive magic bullet for any of this?
Well, I think a vaccine will be our magic bullet, right? To really help protect as many people as possible. I don't think there's going to be one magic bullet. I think it's going to be a combination of strategies, both as we heard, different interventions for protection or prevention and better treatments. And then serologic testing. The combination of things will be able to, do better than we have been in the last several months.
Yeah, along those lines, I'm thinking about everything that you guys in each of your silo, or I shouldn't say silo, but in your fields, what are the boots on the ground needs that you've seen, I guess, that you've encountered? Well, and what I mean by that is when we talked a little bit, we talked about facilities being a limiting factor, by way of example. So what other needs have you actually seen that have affected your ability to do more research or to delve more into the problems of the pandemic?
Yeah, well, to do more research, I do think boots on the ground. We've had incredible infrastructure through the Clinical Translational Science Institute, but more study coordinators, I think having investigators, people who are trained in clinical investigation and having training programs that support the development of people in careers that want to do this kind of research. It's incredibly important underpinning of being able to do this. We basically, in infectious diseases, got all our fellows, all of our faculty just dropped what they were doing and got to work on this. And I think that was the case in transfusion medicine as well. Just the outpouring of support and time from the residents and fellows in that program have been just so valuable. And these are things that people take for granted in terms of how they're funded and supported, but they're so important to being able to respond like this.
I would just add that, you know, the state has taken quite an economic hit during the pandemic. And as a UC system, you know, that translates to support and funding of each of the UC, campuses. So, you know, collaborations with industry as well as, you know, active involvement of people that are part of our donor community is really important for us to be able to continue working at the high level of research that we have in the past. I will also say it highlights why it's also important for us to figure out a way of empowering other medical facilities that are within our local regional area. You know, the county hospitals and others are also going to need to provide outstanding care to the population, because we're all affected, you know, by the lowest common denominator of our strategy for the pandemic. So I think, you know, although we didn't emphasize it in this particular seminar, issues in terms of how we can get it out to a more diverse community, mental health issues that are also, you know, clinical questions, but not clinical trials per se, but an emphasis of a lot of our other researchers, how people perceive risk, their behaviors around perceived risk, the policies, the data that supports those policies. There's active research in all those areas that's happening at UCLA that I think is going to be also very important in helping us navigate outside of this pandemic.
Along those lines, how would you say that the pandemic has affected, you know, funding?
Wait,
Pre-COVID, post-COVID.
I would just like to add on to Arash's previous comments in terms of the equity of medicine. I feel that there have been patients who have come to UCLA to be able to participate in some of these trials. And I've received calls from family members of patients at other hospitals looking to get enrolled or receive plasma, convalescent plasma. And so the knowledge base or the comfort or the infrastructure that Dr. Currier spoke to doesn't exist necessarily in the community hospitals. And if there was an opportunity to expand this research infrastructure, both from an educational standpoint and a patient access standpoint into our larger community, I think that would be very powerful, both in doing these studies and improving treatment for patients, but both in as well as in access to care.
We have a question. Dr. Fauci has indicated that the US should have 100 million doses of one candidate COVID-19 vaccine by the end of the year. And by the beginning of 2021, hope to have a couple 100 million doses. Do you agree with this estimate and timeline?
Nash, what do you think?
I honestly don't know where that quote came from, and I think it's optimistic, and I'm an optimist too. So cheers. But at the same time, you know, we know that making vaccines is just part of a long process. To develop a product in the lab that can neutralize the virus or produce the kind of immunity that neutralizes viruses requires then testing. You have to put it into different patient populations and make sure that they are able to generate enough antibodies and T-cell immunity to actually protect themselves. Those things are not easy. Vaccine development thus far has been roadblock after roadblock after roadblock in terms of development. For example, the bacteria that cause ear infections and pneumonias and meningitis in children called Streptococcus pneumoniae there was a vaccine developed in 2000 for these children. It took 10 years for the next iteration of that vaccine to add just a few more bacteria into that vaccine. It is extraordinarily difficult to make vaccines right now. The infrastructure is in place. Of course, you heard about 150 vaccines being developed, but how many of them will actually make it through all the different hurdles along the way, regulatory hurdles, immunological hurdles, other hurdles, manufacturing hurdles, and out into the community is not clear. I think, you know, again, I'm an optimist at heart, and I really do hope that we have 10 vaccines available. But beyond 100 million doses, remember there are 7 billion people in this world. We have to really think about global access. Whatever we do, we have to think about how to really get it out to the rest of the world and not just in the United States. So as much as 100 million sounds like a lot, it's probably not enough.
Yeah, I mean, I would add to that though. There are plans in place right now to launch at least four studies with 30,000 people each that are going to start rolling out in July and August and September of this year. And as these vaccines are being tested, they're taking some risk to manufacture them in case they're effective. So this is a vaccine development effort like we have never seen before. And so I think that, you know, everyone is really working really hard to try to see if this reality will occur. And I think there's a chance we could make it, and we just have to try.
Okay. Another question that cropped up was, given that so much attention has been given to COVID, what do we know about the nature of pediatric inflammatory syndrome, and what response are we seeking in research and clinical communities?
Yeah, I could try to start with that. The patients who've had this immune response, this exaggerated immune response after the infection, have fortunately thus far been rare. It's been about 100 to 200 patients in New York, and considering the context of about 1.7 million children in New York City, that represents a relatively tiny fraction of children who undergo this reaction. It's probably not the virus itself, but the immune response that's engendered after the virus that creates this kind of inflammatory state. And to be clear, other viruses and other bacterial infections engender similar immune responses in children. This is not completely out of the blue. There's whole fields of us who think about patients who undergo severe inflammatory consequences after infections. There are patients in our ICU right now who have these kinds of responses to other viruses and other infections. So this is not a totally unknown for us. We're treating this like a rare disease because it is an extremely rare consequence of infection. We think that many of the patients who get this kind of hyper-exaggerated immune response after an infection have some genetic dysregulation of their immune system. Something that normally tunes the immune system up and tunes it back down again after an infection comes and goes is broken. And it allows these patients to have an exaggerated post-infectious complication. To understand the mechanisms as to how the immune system does that kind of exaggerated immunity is too much to explain in this kind of seminar. But I can tell you there's at least 30 genes that have been identified in a variety of different pathways, immunological pathways, that create exactly this kind of exaggerated immune response. So we expect in these kids with the hyperinflammatory state to have mutations in those genes and pathways, and maybe some new ones too. And so every patient in the world who has this kind of response right now is getting genome sequencing through a coalition effort that's being run out of the NIH and out of Rockefeller University. I'm part of that group. I'm happy to talk to people offline about it if they're interested in this particular condition. And I would like to really raise attention to the Pediatrics Grand Rounds speaker this Friday morning will be Dr. Moshe Arditi from Cedars-Sinai, who is going to speak for an hour about this particular immune consequence.
There is a question with regards to maximizing outreach. And this, I think, is for Dr. Ziman. What factors assist in bringing treatment, such as hyperimmune plasma, to the broader community? What do you envision as a viable approach for maximizing outreach?
Can you repeat the second part of that question? I didn't.
Sure. What do you envision as a viable approach for maximizing outreach? In terms of delivery, I suspect, of hyperimmune plasma to a broader community.
So I think as the pandemic has continued and we continue to have engagement of our community in donating plasma, our inventory of plasma will increase, and that in and of itself will help support the cause. I think initially as donor centers and larger centers like the American Red Cross started to build up their programs, there was such demand that they didn't, and in some instances still don't have enough product to meet the need. But I also think that there's a component of educating clinicians that this is an option. So the Mayo Expanded Access Program really made it easy. It made it easy for physicians, if they were aware of it, to enroll in this program and make the product available, but it requires the physician that's caring for the patient to be aware, the blood bank at that hospital to be aware of how to request the product and bring it into inventory. And so there still are some logistics, even though it's been simplified, that require knowledge. And that transfer of knowledge so that people know that it is widely available, I think, is one thing that we could do better or help support.
I think it's also a testament to the leadership of certain people at UCLA, Dr. Ziman in particular, who's involved at a state level with people across the other UC campuses to try to figure out how we can create a common platform that might point patients to being able to know where to go, to be able to get these products, and also their providers. And I think, you know, that's an important component of, I think, the leadership that our institution has demonstrated.
As we get into the last 5 minutes of the webinar, I'd love for to go around to each panelist and just talk about what else we can do. You know, people are talking about a second wave and things may or may not get better. But as you're there doing your very, very important research and touching on what Manish said, it's going to take a long time to get to a vaccine or to even a cure. What can the community do to help support the work that's happening in your world? We can start with Judith.
Okay, thanks. Yeah, I mean, I feel like we got out of the gate at a sprint here, and we're all like running as fast as we can. And for people that are runners, you know, you go out of the gate and you start running really fast, and then you realize that you're not in a 5K or a 10K, but you're in a marathon or an ultra marathon, and you got to keep this going for a while. And so I think that, you know, that is, I worry about that. I worry about people burning out, and as things kind of come back, other things kind of come back online, they lose interest, and the initial adrenaline is starting to fade. So I think that, you know, continued support for this work is going to be so vital, and the resources to make it happen. Our study coordinators have been working 7 days a week, and really minimal breaks. I mean, I get emails at 8, 9, 10 o'clock at night of forms to sign and things to look at. And I think just really being able to expand our workforce to be dedicated for this, you know, it doesn't, I just would be remiss not to mention that this June 5th is the, you know, anniversary of the first cases of AIDS that were reported in Los Angeles at UCLA in 1981, 39 years ago. And you think about the effort that it took to come up with effective treatments for HIV and how long it took. We're really building on the backbone and the infrastructure that followed that response to that disease, but it's not, you know, it isn't going to be a quick one, and without really dedicated resources, we're not going to be able to, you know, do as well as I think we've been able to do for this initial period of time.
Thank you. Alyssa?
I think Dr. Currier really summed it up. I think the resources, the engagement, the and specifically with convalescent plasma, as people continue to unfortunately get sick with COVID-19 and they recover, their willingness to donate, whether to donate plasma if they qualify, or to participate in other research studies where we can better understand their immune function or antibody titers in plasma and find better treatments for patients, I think will be essential. So really it's that engagement, whether it's on the research side or the community side, so that we can come together and find, you know, valuable treatments for this disease.
Thank you. And Manish?
Yeah, I'm really excited about a number of the efforts that I'm part of and that I'm following all over the world about trying to understand why some people do so well with this infection and why some people don't, and to try to understand what is the underlying immunological basis and genetic basis of that difference. I think is super fascinating, and it's going to answer questions not only about this disease, which happens to be on a per person level, not a very fatal infection. I'm really heartened by the CDC numbers from last week that suggests that only a fraction of a % of people who get infected actually go on to die. Only a few % of people who get infected need hospitalization. That's heartening. Of course, when you multiply by hundreds of millions or billions of people, it still adds to a lot of people who get sick, and we need to prevent that, of course. But understanding then why that tiny sliver of patients who does go on to getting very sick, why that happens, what goes wrong in the immune system, to me, that's really important for us to learn, because the lesson here is going to be impactful for influenza and for other viruses that take lives and bacterial infections. And I think there's always so much to learn, lessons that we can learn from our patients when they get sick. It sounds crazy, but we're still in a learning phase for a lot of our immunological knowledge. And I think this is an opportunity to really try to get our hands on some really interesting phenomena, try to understand it. And of course, the benefit from that is that we could come up with treatments like these immunoglobulin therapies that I talked about. Those things that protect some people are actually going to be drugs for others. And I think that's really a fascinating part of the immune system in this disease too.
Thank you. And Arash?
I would just say, you know, that our community of researchers and patients and, you know, our broader community in LA came together and I think responded very well to this pandemic. And it's a testament to kind of the lower numbers in our region compared to other areas in the country. And it shows what can be done when everyone comes together, focus on a common problem. And given what's happening on a day-to-day basis in our community that we see on the news, I think it's an inspiration in terms of what we can do when we put our minds to it. And hopefully the innovations and how we learn to kind of move more nimbly and rapidly in terms of doing clinical research for COVID, we'll be able to use for the many years of additional COVID research that we do, but for all of the non-COVID research that we do as well. And I think this will just make us stronger and make us more competitive in terms of performing clinical research.
Thank you so much. Thank you, everyone. I'd like to turn it back to Mark for some final comments.
Yes, I'd like to thank the panelists today because really informative, inspirational, and highly interactive. This is exactly what we're driving with LABEST, and especially reflective of the UCLA culture. So thank you again for our panel, especially taking time out during your busy schedules to address the pandemic and enlightening us today. And also would like to briefly just thank our sponsors of LABEST because they are part of this community. And again, the way we get new patients or therapies to patients, such as the Hyperimmune plasma, is through industry and academic engagement. So again, would like to thank them at this point and turn it back over to Marvy. Okay.
Thank you again for all your time to all of our researchers. We really appreciate it. And thank you so much to all the folks who attended. This webinar will be recorded and sent into our website, so you can check it out then. And everyone have a great day and stay safe. Thank you so much.
Thank you.
Bye-bye.
Thank you.